Good afternoon. It's with great pleasure that I present our work on systems biology and neuropsychiatric disorders with the subtitle Pathways to Precision Medicine. Basically, I'm going to talk to you about how we can begin to use functional genomics to organize and understand complex neuropsychiatric conditions such as autism, which is the focus of our work. None of the disclosures that I have above are related to the material that are going to be presented today. The outline of our work is to understand how systems biology and integrative genomics reveal convergent mechanisms in psychiatric disorders, again in autism. I have four parts to this, of course, in the 10 minutes, each is going to be quite short. First, I'm going to show that we can use a network framework. Basically, instead of looking at each gene in isolation, look at how genes are related to each other in detail. And that will, in turn, help us define a signature of autism spectrum disorders in brain, understand convergence of risk genes, permit disorders across comparisons, and identify drugs. Advances in autism genetics have been extraordinary. In 2008, there was a handful of genes, little mechanistic understanding, and most pharmaceutical companies were withdrawing from the CNS drug development in general, including in psychiatric disorders like autism. But 8 years later, let's jump ahead, we already have more than a dozen genes. We have functional genomic analyses that are pulling those genes together and showing that they're working in very specific pathways. We have more than 200 candidate genes at that point, clear mechanistic models, many advances, and major drug development efforts in autism ongoing. And just to show that in a little bit more detail, by that time, by 2016, what we had understood was that part of autism, perhaps up to 20%, was due to large effect size, mostly de novo, but sometimes inherited mutations, and none of which account for more than 1% of autism. But together, one can look at autism as a collection of rare disorders. Of course, there's also common inherited contributions and a big part that was unaccounted for that we think is due to inherited rare variation as well. But again, this notion of at least a substantial portion of autism due to large effect mutations that can be identified. So the challenge is that advances in genetics and genomics now make it possible to identify genes. And in fact, we had 200 candidate genes now in 2016. Now we have 200 high confidence risk genes. These findings highlight extreme genetic heterogeneity. And so it led to us coining the term the autisms to refer to the myriad genetic causes. But individual genes don't act alone, they act in pathways and networks. So we need to use systems biology approaches to identify these and translate this into mechanism. And again, our framing is we're moving from genetic variation, which is a causal anchor, through these other levels of analysis, DNA to mRNA to protein to synapse to circuit to behavior to brain. But at some level, we're measuring mRNA and using the mRNA to make inferences about function and connect these other levels. But we need to go in both directions, from brain structure, function, and behavior backwards, and from genetic variation forwards to begin to connect these levels. And rather than looking at one gene at a time, shown here on the bottom, we look at groups of genes that are co-expressed or co-regulated that we can identify using these network methods. We identify clusters, groups, or modules of genes within the network that act together and that represent fundamental aspects of biology. And some of the key references are listed below. So despite extensive genetic heterogeneity, there is a convergence of pattern of molecular pathology, not microscopic, but measuring the mRNA, both using microarrays and RNA sequencing. We find that 2/3 of cases show upregulation of microglia and astrocyte genes, downregulation of neuronal genes involved in vesicle transport and synaptic signaling. And we also find we had 8 cases of duplication 15q, which is a major gene form of autism, and that shares the same pattern. When we look to see where the causal risk genes are they in the glia or neurons? They're acting in the neuronal component predominantly. Then more recently in 2018, with almost 2,000 cases and controls across schizophrenia, bipolar disease, and autism, we massively refined these transcriptional networks and have been able to pinpoint very specifically what cell types and what pathways are involved. So we have strong evidence for convergent molecular pathology, and we've used these approaches to compare across disorders to identify what's specific to autism and what's shared across others. Now that's going back from the brain towards molecular. How about from genes? Do the risk genes themselves converge in any processes, time point or developmental process? So here what we do is we take large effect de novo mutations, smaller effect, shared inherited rare mutations, and even polygenic risk. And we ask, does it enrich in specific modules or processes that occur during normal neurotypical brain development? And the answer to that is yes, of course, that autism risk genes map onto specific pathways and developmental networks. Again, we published this first in 2013. Others have replicated it. And in 2019, we extended this substantially to include inherited risk in the paper published in Cell. But we find that autism risk genes from multiple sources converge on prenatal neurodevelopmental processes. They're linked by regulation of these processes at the transcriptional and translational levels. We find that it involves specific cell types, that is implicating long-range projection circuits during the development of cortical glutamatergic projection neurons. When we compare autism to genes across that cause intellectual disability alone, we find that these patterns really distinguish them in that intellectual disability genes, while enriched in a few of the same pathways, are much more broadly distributed and not as concentrated at this period and in this neuronal subtype. This does not mean that autism is specific to glutamatergic neuron. We know that there are genes expressed in inhibitory neurons and more broadly across neurons. But if we want to ask where they converge mostly, where's the evidence? It's there. So although there's a broad effect, there's also some specificity. So what all the genetics and all the systems biology has taught us to this point is, number one, the genetic data are at the population level. We're identifying genes involved in autism in the population. We need to know what comprises disease risk in the individual. So in other words, is it enough to have one large de novo mutation? Will that just give you epilepsy and intellectual disability, and you need polygenic risk or oligogenic risk on top of that? Are there distinct subgroups of disease, so distinct pathways that are kind of non-overlapping? And these questions can be answered in patients by whole genome sequencing at one level. Once we sequence enough patients, we'll be able to ask and answer this question. And to understand the effects of a mutation in an individual, we need rigorous statistical analyses in large populations. But in the meantime, what can we do with these data? Can we use them to find drugs? Can we use any of these transcriptional patterns that we've identified in brain to help us discover new drugs? And the answer is yes, I believe so, although this remains to be proven. So we can use gene expression in disease models or patient tissue, measured using microarray or RNA sequencing, doesn't matter. And we can map them onto something called the Broad Connectivity Map, which is now called the CLUE resource, where they've given drugs to cells and they have expression profiles mapped onto each drug. We can match those patterns and identify drugs that reverse the disease pattern. And then we can test those drugs in model systems. Now, we've used this approach and we have several proofs of principle. We've shown that we can identify drugs that promote regeneration in the central nervous system, that inhibit neurodegeneration related to Alzheimer's disease and other dementias that involve tau. And most recently, we can identify compounds that modify microglia neuroinflammatory function, again, based on taking gene expression patterns, mapping them to drugs, predicting what they're going to do, and showing that the drug does what it's predicted to do. Unfortunately, in autism yet, and in other psychiatric disorders, we don't know what that other phenotype is. We don't know whether we want more synaptic activity, less synaptic activity, more inhibitory neurons, etc. But once we know that, I believe that we can begin to use this approach to identify drugs that can reverse patterns. And this is hope for the future. So in conclusion, we're on the threshold of identifying hundreds of causal genetic factors for most neuropsychiatric disorders. The next challenge is to understand their function. Network approaches provide an unbiased systems-level framework for integrating genomic data, and that these integrative genomics methods and network analyses provide an unbiased genome-wide guide for mechanistic and therapeutic studies in model systems, to identify biomarkers, and for drug screening, as I've shown you. I have to thank all of our funding sources, Takeda Pharmaceuticals, the NIH, Larry Hillblum Foundation, the Adelson Medical Research Foundation, Bright Focus, and the Paul G. Allen Frontiers Program. I have many collaborators who have contributed to this that are shown here. Thank you so much for your attention.